So, welcome to this series on tuberculosis. And as you know, the government is trying to eradicate tuberculosis, end tuberculosis, but I don't think it is going out anywhere soon. So, I feel it is a good idea that all of you learn about tuberculosis, TB as much as you can. It shows up in almost every subject, in almost every examination. And that is why it is a good idea to have a place from where you can read tuberculosis in a sequential manner. So first we'll be going with the second professional subjects, seeing the microbiology, then the pathology, then the pharmacology of tuberculosis. Then we'll move on to the core medicine and community medicine part of it. And finally, we'll see tuberculosis in various small areas, such as where it comes up in obstetrics and gynecology, surgery, pediatrics, small nuances in orthopedics, ophthalmology, and wherever it is required. So let's start off first with our lecture on microbiology. That is the most important lecture where we'll understand the bacteria. And the more you understand about Mycobacterium tuberculosis, the more you'll understand why we give something to treat it, why it causes the diseases it causes. So this is perhaps a very important lecture. I am Dr. Chaitanya. I've done my MBBS from AIMS. I'm currently doing my MD medicine from Jaipur. So today we'll discuss what is the Mycobacterium tuberculosis complex. When you say tuberculosis, what exactly are you talking about? We'll go a little bit into its details of its cell wall, its structure that is important for a microbiology perspective. Then how do we stain it, how do we culture it, and how do we identify it? These are often asked, especially in your professional examination for microbiology. And the part related to its culture, growth, identification is very, very important clinically as well. So when you talk about tuberculosis, you are usually referring to as the Mycobacterium tuberculosis complex. So remember one thing, that if you have to call it a, you usually do not use a gram stain. You usually use a Ziehl Neelsen stain for the tuberculosis organism. But usually, whenever you subject anything to a gram stain, something can be weak gram positive or weak gram negative also. So, tuberculous mycobacteria, by their default setting, are gram positive rods. They are gram positive rods. So, like staph, like streptococcus, they are gram positive, but they are rod shaped bacteria, okay, which have these dimensions. The important thing is that initially, when we started seeing acid fast bacilli, we labeled all of them as a Mycobacterium tuberculosis complex. It was only later when we learned about genetics and we did molecular epidemiology by the method of genotyping, we realized there are multiple things that are included in the Mycobacterium tuberculosis complex. The most common cause of tuberculosis, and wherever we talk about TB, we are referring to it as Mycobacterium tuberculosis. But it is important to realize that the drugs we use to treat it are HRZE, in which you have an important drug, pyrazinamide. So in Mycobacterium tuberculosis complex, you have other mycobacteria such as Mycobacterium bovis that is very, very intrinsically resistant to pyrazinamide. So there is no role of giving pyrazinamide, which is a very toxic drug in case you have a Mycobacterium bovis infection. And how it usually spreads is through unpasteurized milk. The spread is through unpasteurized milk. So if you take milk which has not, and it usually causes tuberculosis in cattle, but it can also be transmitted to humans through this. And the main important thing, the main takeaway from this slide is that pyrazinamide does not work for it. Also, this is the very strain that is used for the tuberculosis vaccine, that is the BCG vaccine. So, if suppose that also you are told that has caused tuberculosis, there is no role of using pyrazinamide in this. So, it only matters. So, laboratory will report it as an MTB complex. Only when it is bovis is it pyrazinamide resistant. There are other strains in the tuberculosis complex such as Africanum, Capri, Microti, right? Other small strains. One or two important points about them. Africanum, it is mainly causing TB in the West African countries, and it is less virulent as compared to the main tuberculosis, right? All of these others are very, very rare human infections. Apart from this, you have non-tuberculous mycobacteria that we will talk about later. Okay, so non-tuberculous mycobacteria are apart from all of this that is included in the M tuberculosis complex. Other thing, when we started doing this molecular epidemiology genotyping, we saw certain lineages. We labeled these lineages from L1 to L7, and the modern lineages that you see that are causing tuberculosis infection currently are from L2 to L4. L2, L3 and L4 are the modern widely distributed ones, and particularly the L2 variant, which is seen more commonly, which was first seen from Beijing, is a very, very virulent strain. So L2 is a hyper virulent or a hyper virulent strain called as the Beijing strain. So these are certain single liner questions that can sometimes be asked to you, and they are important. But my main idea of telling you about this slide was pyrazinamide resistance in Mycobacterium bovis. Right. So we move on to the next point. So let's talk a little bit about non-tuberculous mycobacteria. So remember we talked about uh, the MTB complex, in which you should remember M bovis. It is not a NTM. Okay, do not call M bovis a non-tuberculous mycobacteria. It is a tuberculous mycobacteria. When we talk about non-tuberculous mycobacteria, the important thing to remember, which can be a single liner question, is there is something called as the MPT64 antigen. So MPT64 antigen testing we do, that will be positive for the tuberculosis complex, but it will be negative for non-tuberculous mycobacteria. This can be a single one-liner question that can be asked to you. It has been previously asked. Now, one classification is there that is known as the Runyon's classification, that is important for non-tuberculous mycobacteria, and we'll see a few points regarding non-tuberculous mycobacteria that are important. So you can see this image where you can see multiple acid-fast bacilli. Usually it will happen in the setting of an immunocompromised patient. They will tell you that there is a patient who is having HIV or some other immunocompromised state. The AFB will be negative, maybe the culture is also negative, but the clinical manifestations look similar. And they can ask you what microorganism are you suspecting, and your most common answer will be MAC, Mycobacterium avium intracellular complex. Okay, so the Runyon classification for non-tuberculous mycobacteria talks about pigment production. So these non-tuberculous mycobacteria produce pigments. Some of them produce pigments only in light. They are called as photochromogens. Some of them produce pigments both in the light and the dark. So because they produce pigments in the dark, they are called as scotochromogens. Don't think they produce pigments only in the dark. They produce pigments both in light and dark. Non-photochromogens do not produce pigments, and your MAC is a non-photochromogen. And finally, you have rapid growers. These are organisms which grow within 1 week. So Runyon gave this classification for these organisms. Now, out of these, the important ones you should know, one is Mycobacterium marinum. So marinum, as you can make out, the name marinum tells you marine means something related to swimming or something related to the marine life. So they are common in swimming pools or fish tanks, and fish handlers get this disease of their hands. So this is also, the disease is also called as fish tank granuloma. So anything related to swimming pool or fish tank, you can mark photochromogen marinum, right? That is a marinum. Other ones, bursitis, asiatum, simium, canasi, ginavens, these are all photochromogens. You can just remember the names. Suppose it comes in your exam, to mark which of this is a or a match the column kind of a question. Next, scotochromogens. So in the dark. So how I like to remember this is that your scrotum remains in the dark. It is not exposed to sunlight, correct? So scrofulatium. Okay, it sounds kind of like scrotum. It's a bad way to remember it, but whatever helps you. So scrofulatium, it causes cervical lymphadenopathy in children. It's very, very important. So this is also a potential question. So from the photochromogens, you remember about marinum. From the scotochromogens, you remember about scrofulatium, right? Then in tap water, there is something called as the Mycobacterium gordonii. It is found in tap water. So that is another mycobacterium to remember. Right. Then you move on to non-photochromogens, and perhaps this is the most important group, and we'll talk about it in detail, its therapy and all also. So the Mycobacterium avium intracellular complex, also called as the Pattibacillus, is the most important organism here, and this can be a very easy question to ask, that which Runyon group does it belong to, right? Then in hospital water, you find Xenopi. Ulcerans causes a disease called as the Buruli ulcer, and M para TB is also a non-photochromogen. Finally, rapid growers. Here you should remember one important one, that is Mycobacterium fortuitum, that is seen in post-trauma patients. So patients who have suffered trauma, they may have not mental trauma. From patients who have suffered physical trauma, you will get M fortuitum, right? Other ones, Chiloni, Smegmatis, abscesses, these are all rapid growers in the Runyon classification. So a question you can get from this. So this we have talked about the microbiological classification and division of both tuberculous and non-tuberculous mycobacteria. Now let's move on to the structure. The structure is very important because in the third video we'll talk about pharmacology. In lecture 1C we'll talk about pharmacology, where I'll throw terms at you like mycolic acid inhibitor, right? So one thing you keep in your mind and you ingrain even if you don't remember anything, the reason why Mycobacterium tuberculosis comes is there is something in its cell wall called as mycolic acid that gives it the term Mycobacterium, right? So mycolic acid is a very, very important part of the cell wall, and all the specific therapy that we have, specific therapy, right? Of course, we are using fluoroquinolones and other things, but specific things where you can target these mycobacteria are based on this mycolic acid in its cell wall. There are other drug targets, but the cell wall is a very, very important target. So you just have a look at the structure, and it is the most important slide in this section. The inner membrane, so inside in innermost you have a cell membrane, that is a phospholipid bilayer, universal, present in all organisms, all of that, right? The next layer, it's remember it's a gram positive bacteria, it's a gram positive rod. So just like all other bacteria, this also has its peptidoglycan layer. Peptidoglycan layer of the cell wall is present, all right? That is the layer after the cell membrane, but outside of it it has other layers, okay? The next layer is made up of some sugars, okay, and these sugars are arabinin and galactan, okay, arabinin and galactan. So there is an arabinogalactan layer. The next layer is a arabinogalactan layer, and this is also a potential drug target. So you should remember second layer is an arabinogalactan layer made up of arabinine and galactan, which are some modified sugars. And the last layer outside is a mycolic acid layer. The last layer outside is a mycolic acid layer here, right? And finally, on the top, you have a capsule-like material. Okay. So remember here, whatever drug targets peptidoglycan, so beta lactams, they will work here. Okay, beta lactams, they should work here. But because the penicillin binding proteins are not accessible due to the presence of arabinin, galactan, mycolic acids, drugs like ceftriaxone, drugs like penicillins, they won't work on mycobacterium, even though it is a gram positive rod, because this penicillin binding proteins are not exposed. They are covered by some sugars, they are covered by some mycolic acids. Now, important drugs that I'll just bring here into your mind, of course, I'll talk about this in detail later, is the drug ethambutol. You all know that for TB, the drugs that we give are HRZE. So in that, this E is ethambutol. It acts on this arabinogalactan. Okay, don't think too much. I'll talk about this later in the pharmacology lecture. I just want to drill this concept here that the first drug, that is isoniazid, acts on mycolic acid. So this H here acts on mycolic acid, and the E here acts on ethambutol. The ethambutol acts here on the arabinogalactan layer. Okay, we'll talk about these things in the pharmacology lecture. Okay, so mycolic acids, the important thing to remember is they are very, very long chain fatty acids. Okay, they have approximately 70 to 90 carbon atoms. Your usual stearic acid, palmitic acid, remember they were around 16 carbon, 18 carbon, these kinds of molecules. But mycolic acids are almost 80, 90, 90 carbons, and they are therefore very, very hydrophobic. Okay, so what is this like? So Mycobacterium tuberculosis, it is almost wrapped in a layer of fat. Okay, and therefore all your antibiotics which are water soluble, the enzymes of the host which come to try to destroy it, and even things like detergents, they are bounced off. That is why it is a very resistant organism. That is why it is a very difficult to treat organism. Okay. Now there is something called as the cord factor here that is mentioned in your first aid, and that is the most important virulence factor when we talked about Mycobacterium tuberculosis. So let me write this on a new slide so that you all will remember. So what is the cord factor actually? So cord factor is nothing but a mycolic acid esterified to a hyaluronic. So what we are talking here is that if you have two mycolic acids and they are esterified to a trihallose molecule, a trihallose sugar, this is what is present on the outer surface, and this is what is called as the chord factor. So what you can call this is a dimycolic or a dimycolate molecule esterified to the sugar. So a trihalose dimycolate molecule is the chord factor. Now it is responsible for as a very important virulence factors, and it does very important things. The first thing it does is it contributes to the serpentine cord appearance of the MTB colonies. Okay, so they will appear like a snake like cord. Okay, this snake like cord appearance is due to the cord factor. That is why it is called as the cord factor. Second thing is it activates macrophages and contributes to granuloma formation. It activates macrophages via a special macrophage receptor and causes granuloma formation. Third thing is it causes release of TNF alpha. It causes the release of TNF alpha, and again it helps in granuloma formation. Surface glycopeptides, they also help in, you have surface glycopeptides, and they help in inhibiting phagosome and lysosome fusion. That is a very important thing. That is why it is able to survive intracellularly, right? So you can remember these things, and these are the important points related to the virulence factors of Mycobacterium tuberculosis. Right now, one question that is asked from this cell wall part is that which test for tuberculosis is recommended in HIV patients with a CD4 count less than 100. Okay, in that you detect this arabin and galactin, and you do something called as the urine lamp. So this is mentioned urine lamp test, which is a lateral flow acid test used in patients with HIV and a CD4 count less than 100. So just remember this as a one liner. Okay, I hope you have understood. We'll go to the next point where we'll just summarize what all we have discussed. So peptidoglycan, it is similar to what we have discussed, and we are not able to reach to it directly. Next layer was arabinogalactan, and it is unique. And here what we are looking at is ethambutol as the molecule. Last layer, mycolic acid, which is a very big packing, a lipid wrapping of the mycobacterium, which is very long, has 70 to 90 carbons, and isoniazid tries to block mycolic acid synthesis. All right, and it also protects it from detergents, from host enzymes like host lysozymes, and it forms a permeability barrier. It is also responsible for the acid fast staining characteristics, and that is why nutrient uptake is also slow. So it is a slow growing Mycobacterium tuberculosis is a slow growing bacteria, right? And final thing that we talked about is lipoarabinomanans. They are a component of this glycolipid, and in the urine of tuberculosis patients, they can be detected, right? So there is an antigen test that is approved for advanced HIV with CD4 counts less than 100, and it does not have a good sensitivity in HIV negative patients. So this is a question that has been asked previously. So you should remember about urine lamb test that is useful in urine for detecting TB in patients with advanced HIV, that is those who have a CD4 count less than 100, right? Now, I told you that it is a gram positive, right? And gram positive means it is a weak gram positive, but the main staining that you use is a acid fast staining. So why is it called acid fast? Okay, it means that even if you try to decolorize it using acid alcohol, it still remains stained with carbol fusion. It still remains stained. It does not lose its staining by acid alcohol. That is called as acid fast. Okay. So peptidoglycan. So the concept was that gram stain was made for organisms which had a peptidoglycan cell wall. So even though it has peptidoglycan, here the outer layer is not is ha it outer layer has mycolic acid. So your crystal violet stain of gram stain is not able to penetrate properly. That is why it is a weak gram positive organism, right? So what you should do here is an acid fast staining, where you do is you apply something called as the carbol fusion dye. The carbol fusion dye, it goes inside and it stains the organism. So to send it inside, you can use heat or you can use phenol. Okay, you can use either of these to help the carbol fusion to penetrate, and then you apply a decolorizer to see whether whether the organism is acid fast or not. For non-acid fast organism, the dye will come out and it will get counterstained by your counterstain, which is a blue or a green stain. Whereas mycobacteria, their characteristic is that mycolic acids are present, so they form a complex with the dye and it remains even after doing decolorization. So these organisms will appear red or pink. Okay. So important thing is the dye here is the carbol fusion. The mordant here is either heat or phenol, depending on whether you are using hot or cold method, which is Ziehl Neelsen and modified Ziehl Neelsen or Quinoan staining. The decolorizer is acid alcohol. The counter stain is malachite green, and the result that you get is a bright red or pink rods on a blue green background. And what is responsible for this acid fast nature is the mycolic acids, right? But based on this, you cannot differentiate each of the mycobacteria from each other. Those are the important points you have to remember. Okay, so let's go through this once again in a schematic way. I'll draw all the layers again and I'll try to show you what I'm trying to talk about. So by now you should be able to draw the cell wall from memory. The first layer was gly the first layer was glycolic acid, right? The first layer here that is responsible for all this is the mycolic acid, right? Or sorry, the first layer here is the mycolic acid. The second layer you had your arabinogalactanins, and the third layer was your peptidoglycans. The third layer was peptidoglycans. So had it been just peptidoglycans, what happened? It would get positive gram stain with a crystal violet. So when you try to do a gram stain, the crystal violet stains this layer weakly, but the stain is very unreliable. So you are not able to use it because of the presence of these layers. So what you have to do instead is you have to use carbol fusion. Carbol fusion as the stain. Okay. Now carbol fusion, what it does is it goes inside and it binds, and the mordant that you use is either heat. Okay, that was used previously, heat. The problem with heat, so this was known as the Ziehl-Neelsen method of staining, ZN staining. But the problem with this method is when you use heat, okay, you produce aerosols. It is aerosols and it is a biosafety hazard. The people who are doing this can get infected. So it requires a PSL3 lab, but we have to do it even at a PHC. So the method that we use here is the cold method or the Quinoan's modification, right? In which you use phenol as the mordant, right? Mordant means something which causes the dye to stay, right? So when you put this mordant, the what happens is the red pink color of the carbol fusion, it sticks, right? And then you try to decolorize it. You try to decolorize it with acid alcohol, but that doesn't work. And then you try to put a counter stain, malachite green, but because it is not decolorized, it will not take up malachite green. So the counter stain here is malachite green, which gives the green blue background that you will often see when you are seeing a AFB smear slide. So if you look at this slide here, this blue background is due to the counter stain, and on top of that, the small pink things that you see are the acid fast bacilli. Okay, so that is the principle of acid fast staining. So we'll discuss this a little bit more in detail. One important thing here that is more important for your theory examinations, or in fact your microbiology practicals and your clinics, is what is the optimum sample. So previous recommendations were that you needed three samples. Okay, you needed a early morning sample, you needed a spot sample, then you needed another sample. Now they say you need two samples. One is a spot sample. Next is an early morning sample. The reason for that early morning sample is so that throughout the night, whatever bacilli are there, they will accumulate in the sputum, and when you take an early morning sample, you will get good yield. In children, you can go for a gastric aspirate. Then you can also take bronchoalveolar lavage by doing a PAL, right? These are the things that you can take. For extrapulmonary tuberculosis, you have to take from the site. You have to take from the respective site. For example, if you're suspecting TB meningitis, you need to do it in CSF. The processing of this sputum, sometimes this comes as a single liner. The method uses the modified Petrov's method, where you use 4% NaOH and N-acetylcysteine to dissolve the mucus. So there's a Petrov's method, there's a modified Petrov's method. Basically, you are using sodium hydroxide to dissolve the sputum. That is what is the Petrov's method. It is a method of processing the sputum before doing acid fast staining, right? Now, other things that you have to know, that you can have a sterile sample, CSF, pericardial fluid, synovial fluid, ascitic fluid, where you can look for TB. Then you can have pleural fluid, you can have blood, you can have laryngeal swab, you can have a swab from a sinus, early morning urine or stool. All of these are samples that can be used. And how many ml is required? I've written in this slide, most important things that they have to be collected early in the morning so that all of this is accumulated. For urine, the recommendation is you have to have at least 500 ml specimen, and it should be centrifuged because there is intermittent shedding of the bacilli in the urine. You should take at least three specimens on different days. Okay, so this will be more useful to you when you are practicing as a medicine resident and you are trying to prove tuberculosis in a patient. What is the optimum sample to send? Right. Next, staining methods. We have already discussed this. The hot method is the Ziehl-Neelsen method, in which what you are using is you are using heat. You are using heat, but because this is an aerosol generating procedure, you resort to the Quinones method, where instead of heat, you are using concentrated phenol. Okay, and the sensitivity is the same. You need approximately 10 to the power 4 colony forming units, but the advantage is that you will not have generation of any aerosols. The best staining method is fluorescence microscopy, where you use a specialized dye called as the auramine dye or the auramine rhodamine dye. Here, heat is not required. The organisms will show yellow-green fluorescence on a dark background. Okay. So here the sensitivity is increased. Remember, here you needed at least 10,000 colony forming units. So it is 10 times more sensitive. It needs only 1,000 colony forming units, that is 10 raised to power 3. Okay. It is faster also. You don't have to do all the staining process as you were doing heating and all of that. And you can screen more slides per hour. Okay. But again, disadvantage, just like the ZN method is, you cannot distinguish the Mycobacterium tuberculosis from non-tuberculous mycobacteria. Okay. So, which is the more sensitive staining method if you are asked? It is the auramine rhodamine method. What is the sensitivity for Ziehl-Neelsen stain? 10,000. What is the sensitivity of the fluorescence method? It is 1,000. And why are we using cold, not hot? It is because this generates aerosols and it requires a BSL-3 setting, and it can be possibly infectious. Okay, the difference is here you are using concentrated phenol as the mordant. And here, can you diagnose Mycobacterium tuberculosis? No. You can only tell that acid-fast bacilli are present. You cannot distinguish between species here. So that is very, very important thing. Like I said, sputum samples you have to use. You can use a spot and an early morning, and the yield will be lower when it is a HIV positive patient or someone with an extra pulmonary tuberculosis, because you have paucibacillary disease here. Previously, they used to ask a question from RNTCP, which is no longer part of anything, that if there are no AFB, you report it as negative. Then scanty means that there are less than 10 acid fast bacilli in 100 fields. 1 plus means there are 10 to 99 bacilli in the 100 fields. 2 plus was there are less than 10 bacilli per field, and if there are more than 10 bacilli per field, that was 3 plus. That is no longer asked. It will be there in some old questions, but you need not remember. Okay. So this is the important thing. Now, recommendations are that two samples of sputum are sufficient. A early morning sample and a spot sample. Previously, three samples were required: spot, early morning, and spot. So if you see an old question, just ignore it. Okay. So in India, what we are following is NTEP, and two samples are required. Okay. You can have a look at this image. It is showing you fluorescence in the auramine rhodamine stain. Coming to growth and culture. After staining methods, we come to growth and culture of Mycobacterium tuberculosis. So one thing you should realize is that it is very, very difficult to prove with culture. Culture is the gold standard for any microbiological diagnosis. Fox principles tell you that if you are able to culture an organism from somewhere, it means that there was a live organism that was causing the disease. So culture is definitely the gold standard for each diagnosis. Okay. But the problem with tuberculosis, like I told you, is that it has mycolic acid in its cell wall. Okay, this mycolic acid, what it does is it restricts nutrient intake. Mycolic acid, okay, it helps to prevent so that there is no antibiotics do not come inside, enzymes do not come inside, detergents do not come inside. But the disadvantage is, or the advantage is, that nutrients are also taken up slowly. Nutrients are taken up slowly. Because of that, it is a slow growing organism. It grows slowly, so it does not show positive cultures. So like blood cultures and all, you will get positive within 7 hours, 10 hours, 12 hours. The reason for that is something called as the generation time, right? E. coli, which is isolated in blood culture, and I have seen many patients who have a positive E. coli blood culture. The generation time is only 20 minutes. So you can typically get a culture positive within 1 or 2 days. Mycobacterium tuberculosis, instead of 20 minutes, almost 50 times more generation time is present. It has a generation time of almost 14 to 15 hours. So to double, it takes almost this much time. So if you want to culture it on a solid medium, it will take approximately 2 to 8 weeks. Frankly, I have sent many cultures. I have sent many cultures for Mycobacterium tuberculosis, but by the time we get culture positive, the patient is already discharged and has already completed the intensive phase of therapy. So it is a very slow method. One possibility to reduce that is to use a liquid culture technique where you get culture in 1 to 3 weeks. But still, the time here is in weeks, not in hours or days. That is a disadvantage of culture, but definitely it is the gold standard technique. Some important points that you have to understand. So, because of this reason, because of this very reason, because E. coli divides so fast, right? You can give antibiotics and within 5 to 7 days you will be able to treat it. But because tuberculosis divides every day or divides every 20 hours, to kill the same number of generations, you will take months. That is the whole reason why tuberculosis treatment takes almost 6 months. Okay. Now, one more thing is that in because of this, when you look at a TB colony, there are certain bacilli which are actively dividing. Okay, they are actively dividing. They will be outside the cells. Then there will be some bacilli which are dormant, lying inside the cells, and there are some bacilli which are closed, which are in closed areas surrounded by caseating necrosis around them, and they are within those places. They are latent. They are non-replicating bacilli. So because of this, you need different types of drugs. That is why you need multi-drug therapy to act on tuberculosis. So these growth characteristics help you understand more things when you think about the pharmacology and the medicine part of this. Now let's come to the growth medium, that is coming back to the microbiology. So you have the Lowenstein Jensen medium, which is used for its culture. The LJ medium. The LJ medium. I hope you have seen how the LJ medium looks like. So it is an agar based medium which has egg. It contains egg and it contains malachite green. So malachite green is important here. Why? Because it prevents normal flora from clinical specimens. So if you are telling me that the growth time of E. coli is 20 minutes and my sputum has some flora like E. coli, so the culture medium has to prevent the growth of that normal flora to allow the tuberculosis to grow. So malachite green in LG medium also gives its color, the greenish color, but it is bacteriostatic for almost all bacteria. So it stops the growth of all other bacteria, but because of the waxy cell wall, mycobacteria are able to grow. So that is why it is a very, very important constituent of the Lowenstein-Jenstein medium. Other things that it contains are, it contains glycerol. So if it contains glycerol, Mycobacterium tuberculosis will grow on it. If you want to grow Mycobacterium bovis, you need to provide pyruvate. Then other things are asparagine, mineral salt solution, and optimum temperature is 37 degrees Celsius, pH of 6.4 to 7. So remember, your body has a temperature of 37. It has a pH of around 7. So all the optimum conditions are present in your body. Then it is an obligate aerobe. So it is a aerobic culture medium. That's why it's seen very commonly in your lungs, right? And how are the colonies? They are described as rough, buff, dry, cauliflower-like colonies. Rough, buff. Okay, you should remember the words rough, buff when you are describing Mycobacterium tuberculosis. Okay, and here you need lesser organisms that you need for smear. So for a smear to be positive, you need 10,000 if you are doing an acid fast staining with a ZN method. But here, even if it is smear negative and the organisms are viable, you will get culture. So it is the gold standard for culture based diagnosis, but it takes 2 to 8 weeks. The other alternative that they tried was they tried culturing it in a liquid medium. That was the MGIT, Mycobacterial Growth Inhibition Tube. And there are certain names for it, the Backpack 960 or the 460 TB. These are basically liquid or broth based cultures, right? Some people call the technical name for it is the Becton Dickinson tube. That and all is not required for you. Only thing you have to remember is that when you're talking about broth base, the culture medium they use is the Middlebrook 7H9 plus OADC, which is oleic acid, albumin, dextrose, and catalase. All of this you can find in the notes that I will provide to you. You need not write all of this down. All of this is already there in the notes that will be there in the link for this video, right? The principle here is that there is a O2 sensor, there is a fluorescent O2 sensor. So whenever those organisms grow, which are aerobic organisms, they consume the oxygen in the tube. When the oxygen level goes down in the tube, the oxygen sensor, which is a fluorescent sensor, detects within 1 to 2 weeks that oxygen has been used. And then you can use this for drug sensitivity testing also for all the four first line drugs. And it is FDA approved for this indication, right? Other alternative is the backtech method, where instead of a sensor which is a fluorescent sensor, you use a colorimetric carbon dioxide sensor. Okay, but it is not approved for drug sensitivity testing. So MGIT is approved for both drug sensitivity and identification, whereas the backtech is only approved for identification. Okay, it is faster, it is automated, and you can monitor. It is cheap, so it is having very good sensitivity and specificity. It's a amazing thing, right? So let's have a look at this. So this is the LJ medium with the malachite green that you are able to make out. As I told you, important words are rough, buff, and tough colonies of Mycobacterium tuberculosis. And this is the MGIT tube, and you can see the fluorescence. So in these tubes, oxygen has been consumed. So you can see the fluorescence telling you that it is MTB positive, right? So some other points, some one-liners related to culture medium. So if you add iron, you add for uptake, for differentiation and identification. Pyruvic acid is for enrichment and particularly useful for M. bovis. 5% sodium chloride for certain strain you can add. Then you have a ESP2 system, extra sensing power, which is based on pressure changes. Remember, the O2 sensor was used by MGIT. This is one ESP2 system which is based on pressure changes. Then you have MPT64 antigen detection, which is an immunochromatography based method. And remember, I told you non-tuberculous mycobacteria are MPT64 negative, right? Then you have lysis centrifugation based augmented culture, which can be used for blood. Blood culture will only be useful if you have disseminated tuberculosis, and in that you can use the lysis centrifugation augmented culture methods. All of these are one liner questions which can be asked, but you need not focus on it too much for a theory examination perspective, right? Now let's come to organism identification and drug susceptibility testing. This is something that we have discussed here, that the sputum samples need to be transported as soon as possible. Maximum you can keep the sputum or the CSF sample in fridge for 3 days at 4 degrees. But suppose you have to send these samples, you are at a primary center and you have to send it to a reference laboratory. You should use the CPC medium plus normal saline for couriers. And the method we already talked about is the Petrov's method, where you use sodium hydroxide plus minus N-acetylcysteine if it is the modified Petrov's method. So these things are related to sample storage and transport. Now, organism identification. So suppose you have isolated a culture and that culture is having an organism, or you can do direct organism identification and DST as part of our NTEP program. You want to know that which organism is it and what drugs work for it, right? And the best thing that we have here with us is GeneXpert. GeneXpert for diagnosis of Mycobacterium tuberculosis with rifampicin sensitivity, and that technology is called as the CBNAAT. Okay, there are certain important one-liners here that you need to remember. We'll just go through them quickly. The first thing to remember is that it is based on the principle of polymerase chain reaction, amplification of DNA in a self-contained cartridge. That means that the cartridge does all the job. So it is a semi-automatic process. The PCR that goes in it is called as the hemi-nested real-time PCR. Okay. What it does is it detects Mycobacterium tuberculosis, and at the same time it also detects rifampicin resistance, right? And here it detects two things, right? It detects the presence of the bacteria, and not only that, it also detects the RPOB mutations for rifampicin resistant. So it allows you to immediately start treatment and tells you that okay, it is a rifampicin, it is possibly rifampicin sensitive. And the sensitivity here is further reduced. Remember, we talked about AFB. There the sensitivity was 10,000. For culture, you needed 1,000 colony forming units. Here you almost need 100, right? So here the sensitivity is 131 colony forming units per ml. So you are coming down on even high. So it has a higher sensitivity than culture in picking up organisms. You get a result within 2 hours, and it is approved for patients who are uh, from the respiratory specimens, and it is not approved for identification from culture growth. You have to take it from a respiratory specimen. And in smear positive cases, you have seen a sensitivity of 88%, 67% in smear negative cases. But if you get a positive result, it is 99% specific. They have now made something called as the GeneXpert Ultra, which I have seen being used for CSF samples, where the sensitivity is further decreased. So from you started from 10,000, came to 1,000, this was 100, that is 10. Okay, you need 16 colony forming units. And for paucibacillary disease, GeneXpert Ultra is the new update or the new technique. But it cannot identify all mechanisms of resistance. It is only identifying a specific RPOB B mutation, that is for rifampicin resistant. That also it is only able to identify, but that is good enough for us to start treatment. Now, some important things that are still tested in examinations and are important points to differentiate MTB and M bovis. One is the catalase test. Remember this, I told you in, if you remember this one-liner slide on culture, that you add this, you create wells in the LJ medium, and you do the catalase test, okay, which was done previously, and catalase test was positive for M bovis. That is the important point to remember, that M bovis will show a positive catalase test. Rest of the test will be positive for MTB, right? The niacin test, the nitrate reduction, the pyrazinamidase test, okay, all of these are positive for MTB, but catalase test is positive for M bovis, right? So these things you should remember. And one new method again is the MALDI-TOF, right? That is based on the principle of mass spectrometry, and it also allows rapid identification. It is now available in reference labs, but it is not very relevant for clinical specimens, right? So important things about GeneXpert. First, it detects rifampicin resistant. Okay, so if rifampicin resistance is there, you have to start multi-drug resistant regimen. Then you have to do secondary tests to see what all drugs are resistant, right? Now you have GeneXpert Ultra, which has increased the sensitivity to 16 colony forming units, right? Other things that we talked about, niacin test, pyrazinamidase test, all of these are no longer very relevant. But if you get all of this, just remember catalase test is the only important test that is positive for Mycobacterium bovis, right? And because of a mutation, the PNCA mutation, it has a mutation called as the PNCA. Mycobacterium bovis has a PNCA mutation, so it is resistant to pyrazinamide. That is something that I stressed to you in the first slide also. Okay. Just showing you some important data, some important points. This is sometimes asked to you in a arrange this following in a sequence. The sensitivity of GeneXpert for these samples. So you should remember CSF, it has a 85% sensitivity. Lowest sensitivity is for pleural fluid, that is almost 37%, because it is a very unsterile sample, right? For lymph node, it has a 97% sensitivity. Gastric aspirate also 85%. Smear positive samples almost 100% sensitivity. Smear negative slightly lower, 67%. So these data are variable from variable places, but what you can remember is these pooled estimates. You can remember these estimates, but what you will be asked is that pleural fluid has the least sensitivity. Almost remember here 34% sensitivity for pleural fluid. CSF you can remember 85%. Lymph node 96 to 97%. Gastric aspirate 78 to 85%. Smear positive is 95% and smear negative is 69%. These important data. The exact data are not important. This trend or this table is important. Okay. This was the figure when GeneXpert as a technique was discovered. The article was published in NAGM, right? Rapid diagnosis of tuberculosis with rifampicin resistance. This figure is from that article. It shows you how it is a self-sustained cartridge, that you don't need anything else apart from a small cartridge where you put the sputum and you process the sample. Now they have also made a portable version of it, okay, that is coming in this format. So there are multiple names for this, there are multiple things, but you need not remember all that. This might come up as a image based question, so I'm just showing you a few images, right? So positive test is very useful, negative test does not rule out. GeneXpert Ultra is in a faster time, requires less colonies, 16 colony forming units, and that for that they have added a new category of reporting it called as the trace positivity in GeneXpert Ultra. And this here is GeneXpert Omni, which is the portable model. Okay, Omni, GeneXpert Omni. In India, a lot of places what they're using actually is not the GeneXpert method. They are using a different CBNAAT method, which is called as the TrueNAT. It is called as TrueNAT. Okay, it is by a different company. It is a point of care testing. It also takes around 1 hour. It is based on a chip-based method with a disposable reactor. It is also portable. Okay, and it can be used at periphery hospitals. You have to just maintain it at 40 degrees Celsius. It has different primers that can also be used for non-tuberculous Mycobacterium. It also has a very good sensitivity, 99% in smear positive and 75% in smear negative. Then one more thing is interferon gamma related assay, or the IGRA test. And here in the IGRA test, you are having the concept of quantiferon gold. So it basically sees whether a person is sensitized to Mycobacterium tuberculosis. And the good advantage over MAN2 test is that it is positive. It is not positive in patients who have received the BCG vaccination. So it can differentiate vaccination from exposure, right? Other antigens that form the basis of this IGRA test, ESAT6 and CFP10. You should remember this. This can come as a one-liner: ESAT6 and CFP10, right? And here IGRA, you can also differentiate it, Mycobacterium tuberculosis from other bacteria. Whereas you have a MON2 test where you use 5 units, 0.1 ml intradermal, and you read it 72 hours later. This is one small point. When it is positive, more than 5 mm, 10 millimeter, those cutoffs you will read in pediatrics, right? Then you have a color-based method called as the TK medium. This can also be an image that comes up to you in your INICT examination. So what all things you have for your diagnosis? Microscopy we discussed, Ziehl Neelsen staining, fluorescent microscopy. Other upcoming things which are coming up in the pipeline, there is a TBDX, right? Automated system for smear microscopy that is coming up for microscopy. Culture, we discussed LJ medium culture, liquid culture. What are the things that are coming up as thin layer agar, which also detects resistance, TREC, sensititer, myco, TB, MIC. All these names are important. Okay, TK microcolor. I had shown you the TK microcolor medium. These are coming up for culture based methods. Molecular diagnosis, PCR based methods like GeneXpert, where you're using IS6110, MPB64, 65 kilodalton protein, right? These are the mechanisms behind what are the, so I told you GeneXpert was a hemi-nested PCR. So these are the probes it is looking for. Then ligase chain reaction, LCR, then transcription mediated amplification, line probe assay, and loop mediated isothermal amplification. These are the molecular diagnosis methods. Coming methods are true array, hydra array, Vire MTB, right? Late PCR, near assay, LAMP test, B smart, gene drive, gene POC. All of these systems, they are upcoming, but there can all be a single line question that what is the basis for their use, right? Or in which disease are they used for? Non-molecular test, very commonly used for CSF, ADA more than 10 is suggestive of tuberculosis. And upcoming thing is breath analysis. And one more question that is asked is urine LAM test in HIV, advanced HIV, CD4 count less than 100. Okay. So these were the important points from the diagnostic modalities. Now coming to drug susceptibility testing. Now, drug susceptibility testing is of two types. There is a phenotypic drug susceptibility testing and there is a genotypic drug susceptibility testing. The phenotypic one is the conventional one. There, the method is simple, okay, that you try to grow the organism on a culture plate. When you add certain antimicrobial to it, if it grows, it is sensitive. If it doesn't grow, it is resistant. That is phenotypic drug susceptibility testing. Genotypic, you try to predict it without putting the actual antibiotic, but you try to see the genome of the bacteria because you know which are the genes which confer resistance. Okay, that is the basis of saying phenotypic and genotypic, conventional and molecular. Okay. So the most common one which is used for phenotypic is the MGIT system. Okay, and for first line drugs, so you can take an MGIT tube. In one tube you add isoniazid, in one tube you add rifampicin, in one tube you add pyrazimide, in one tube you add ethambutol, and you can see the MGIT system for first line drugs. And here the principle is microscopic observation of drug susceptibility, right? MODS. And here you can test all of them at a single critical concentration. And you can have either it is sensitive or resistant based on whether fluorescence is present or not present. Then you can do for second line, third line drugs for beta-alanine picomanate in broth microdeletion, where you tell a minimum inhibitory concentration. And here for newer drug susceptibility testing, you are requiring reference labs. And this phenotypic drug susceptibility testing is the gold standard. Why? Because you're actually adding the antimicrobial in vitro and seeing whether it works or not. So it is considered as the gold standard. That again, limitation is same as culture. It is slow, it takes time, it requires the live organisms, and all of this has to be done at a reference laboratory. Okay. Now, genotypic drug susceptibility testing, which is now becoming to be more commonly used. Okay, your GeneXpert is an example of a genotypic drug susceptibility testing where you know that RPOBB gene mutation causes rifampicin resistance. So you can use whole genome sequencing or targeted NGS or line probe assays to look for certain mutations. So if you do a simple line probe assay, which is the common one that we are doing now, you look for rifampicin resistance, you look for isoniazid resistance using CAT-G and INH-A. Okay, and then you can define based on these polymorphisms whether resistance is present or not. Okay, when you have a lot of money, you can do a whole genome sequencing, right? And you can do it very quick. You can get your result within hours to days, and you will still need phenotypic confirmation, but at least it gives you an idea of what therapy you can. The disadvantage is, of course, you don't know many genes which are responsible for resistance, so those might be missed. So phenotypic confirmation is very, very important. Okay. So based on this, you will define whether the organism is drug resistant or whether the organism is sensitive to some drugs, whether it is extensively drug resistant. All of these things you can tell based on this. Okay, so you have Indian reference laboratories which are building these capacities, and this is the future of drug susceptibility testing, right? So conventional methods, I talked to you, these four methods that I've highlighted, they are considered as the gold standard methods. The conventional DST in the absolute concentration method, resistance ratio method, and the proportion method on the Lowenstein-Jensen medium. This is the most important thing when you talk about conventional or phenotypic drug susceptibility testing. Another one is the Backtech 460, right? These are some other techniques, E-test, Alamar Blue, MGIT, Backtech, all of these, right? Genotypic methods, important ones that we use in practicality are line probe assay, right? Then other methods, I'll just go through them. Whole genome sequencing using Sanger's technique, PCR, SSCCP, dideoxy fingerprinting, DNA probe assay, fluorophore plus quencher method, RT-PCR, PCR ELISA, oligonucleotide chips, and whole genome sequencing. So anything related to DNA, these are all genotypic methods, and you can get a one-liner question from this, or you can get a question in your examination related to drug susceptibility testing. These values you need not remember, but remember I told you for drug testing in these tubes, you will have a critical value that you will add to the MGIT tube to see this testing. So these are some of the drug levels that they use, and it is given in textbooks, but this is not really important to remember. But you should remember that there is a fixed critical concentration of drug that is added to the tube, and based on that they tell that whether it is sensitive or resistant. Okay, and the backtech MGIT technique requires the important thing to take away from this is the critical concentration of drug required for the Lowenstein Jensen medium is much more as compared to the liquid cultures. For here example, it is 40, whereas here it is just 1. Okay, so this is something that you can remember. And there are new methods, phenotypic methods for drug susceptibility testing, in which one important concept that is coming up is phage based assays, where you are using the D29 phage for drug susceptibility testing, right? So you grow the organism with M. stegmatis, which is a rapid grower, and you look for lysis plugs. So phage based assays, just remember this word if it comes up in your examination. All of these are super rank deciding questions. Okay, so during my NEET UG time, one of my teachers used to use this word, super rank deciding questions. So these questions you may not remember in your examination, but if you have seen this slide or if you have some idea of these words, they will come to you in your examination. Okay, so you might get a question asking you which phage based assay is used for Mycobacterium tuberculosis. You will get options like D28, D29, D30, and B29. So if you have read this slide, you will know, okay, there was just one D29 phage that was mentioned on this slide. Okay. Other methods, using a bioluminescent enzyme, luciferase reporter assays, which has 100% sensitivity for isoniazid and rifampicin resistant. That is another new method for drug susceptibility testing. Okay. From this slide, just remember D29 phage and luciferase reporter assays. Okay. Now this is line probe assay. This is what is used commonly in our NTEP, and I just wanted to show you what it looks like. So there are multiple amplified targets and they have respective DNA probes. and it uses a chromogen which turns into a purple precipitate, which is which is attached through this biotin and streptavidin, alkaline phosphatase, all these molecules. One chromogen is basically attached to the target and probe. So based on that, it identifies mutations and it screens for rifampicin resistant. So all of these RPOB B mutations. For isoniazid, it screens using CAT-G and INHA. So it basically tells you, so the result will be rifampicin resistant isoniazid sensitive, or rifampicin sensitive isoniazid resistant, or resistant to both of this, based on which all your categories have come up. So since January 2018, universal DST is recommended in the RNTCP and NTP programs. At least rifampicin resistance is screened for all TB patients, right? Now, line probe assay is this method, which is a strip-based method, and you can do it from a smear positive patient. You can get a result within 5 to 6 hours. It detects the Mycobacterium tuberculosis complex, so it tells you that even if it was acid fast, it is not a non-tuberculous mycobacterium. It is an M tuberculosis complex. It does not differentiate between bovis and M tuberculosis, and it also tells you rifampicin and isoniazid setting. Okay. And based on this, you can tell whether it is a wild type or a mutated type, and you do not have to wait for culture. So, what type of method is it? It is a genotype. It is a genotypic method for drug susceptibility testing. Right. These are some very, very important questions that can come in your exam. What are the various gene loci which give resistance to uh, various anti-TB molecules that are used? Okay. First one, rifampicin, RPOBB, you all know. So the important codons here are 516, 526, and 531. And those are the ones that are detected by GeneXpert, and they are the ones detected by GeneXpert and line probe assay. Okay. So what it transcribes is the beta subunit of RNA polymerase, that is the target of rifampicin. Isoniazid, it requires catalyst peroxidase for its activation. So CAT-G is one mechanism of isoniazid resistance. Another resistance mechanism for both isoniazid and ethionamide is INHA, which is the product of which gene is enoyl ACP reductase. So INH and ethionamide have cross resistance. If it is resistant through the INHA mechanism, it will be resistant to both isoniazid and ethionamide because their distal target is the same. That is the enoyl ACP reductase. Another one for isoniazid is oxy-R, right? So all of these are one-liner questions. Streptomycin, RPSL, which is a ribosomal protein. Another one is RRS16S, ribosomal RNA. For fluoroquinolone, it is GYR-A, that is the DNA gyrase. Pyrazinamide, it is PNCA, which is an amidase. And remember, this mutation is present in Mycobacterium bovis. That is why they are intrinsically resistant to pyrazinamide. For ethambutol, you have EMBCAB, right? EMBCAB, EMB cap. Okay, so it is a cap which drives the bacteria away from ethambutol, right? So these are the important things. The PNCA mutation is not detected by most rapid tests. The pyrazinamide detection is actually a very difficult one in a practical setting. Okay. Then for some mutations that are not mentioned here, so for amikacin also you have similar ones. For beta-culin, remember ATPE mutation. Okay, it is a ATP synthase inhibitor. So ATPE mutation or the RV0678, also MMPL5 upregulation, right? So just remember these terms, and this will most definitely be an MCQ. You will not get to write this in a long format. So, and it is fairly easy to memorize. So this table is very, very important. Now, finally, moving on to differentiating one important thing, and it is mainly a US terminology. So, whenever you are reading up to date or Western literature, you will come across two things. One is TB infection, and one is TB disease. TB infection and TB disease. So, whenever they talk about TB infection, they are referring to as latent tuberculosis, that will be mentioned in Indian literature, latent tuberculosis. TB disease is the usual full blown TB active disease that we are referring to. Okay. So what happens when somebody gets tuberculosis is 5% of the people, they will not develop any infection. 90 to 95% of the people will get a TB infection, that is a latent tuberculosis, but these organisms are contained by host immunity. You will get a positive tuberculin sensitivity test. Monto test will be positive, IGRA will be positive because immune response has happened, but they will not get an active disease. But if they take steroids later in their life or become HIV positive later in their life, they will get reactivation, and there is a 5 to 10% reactivation risk in these people, right? And primary tuberculosis disease only happens in 5% of people within the first 1 or 2 years of infection, and it is because their immune response is not able to contain the primary disease. Usually happens in young children and immunocompromised. So here, uh, these are the important categories. Okay, so this is much more important when you are talking about tuberculosis in medicine. But I just wanted to bring the concept of TB infection because it is relevant in the concept of TST and IGRA for you. Okay, let's go ahead to transmission, final topic where I talk to you a little bit about how tuberculosis spreads from person to person and why it is so common in India. So one of the theories is, and one of the very acceptable theories is that because in India people spit a lot, that is why tuberculosis is common in India. It's sad, but it's true that people do spit a lot in India, and that is one of the reasons we are not able to end tuberculosis so far. So transmission of tuberculosis, basically what happens is it has a 1 micrometer droplet, okay, and because it is so small, remember you have PM 2.5 particles, PM 10 particles, so those refer to some microns, and these very small particles, they can reach the terminal alveoli and it is very difficult to get them out, right? That is one important thing. Because they are small, even after a patient goes out of a room, they remain there for several hours. Okay, so that is why it is called as airborne transmission. It is called as airborne transmission. So it is not limited to droplets. These particles do not settle down and they remain airborne for many hours. And even 10 bacilli can initiate infection. So one person, when person coughs, approximately 3000 droplets are released, and it is said that a tuberculosis patient affects almost, it gives the disease to almost 10 to 20 more people during the time period of their tuberculosis infection if anti-tubercular therapy is not started. Okay, so these are the important factors for transmission that you should know about. That is why well ventilated rooms are very important when you are dealing with a tuberculosis patient. And 95% of the transmission is inhalation or airborne based due to inhalation of droplets of cough which are suspended in the air from active TB ingestion. Okay. Now, last one other method is oral method. So nobody is swallowing the sputum of TB patients. This is due to unpasteurized milk. You get M. bovis infection, and this is resistant to pyrazinamide. Uncommon routes, there are documented transmissions in a congenital or neonatal fashion, which are transplacental, but these are relatively rare. This is more common when it is a miliary tuberculosis in a mother, right? Injectable, so it has been described that intravesical, that is in the bladder, if you inject live BCG vaccine, it can cause BCG disease, right? Sexual transmission, direct contact with active genital TB lesions. I don't know why somebody would do that, but this has also been described. But the main transmission, if somebody asks you, it is a airborne transmission that happens for tuberculosis, right? So we discussed the Mycobacterium tuberculosis complex, its structure, the staining, the culture techniques, the identification, and drug susceptibility testing. So these are the important things that we have gone through. I know a little bit of the DST part, culture part might not have reached your head exactly as I intended, but I wanted to keep this short. I wanted to cover it under an hour so that it remains revisable, remains digestible, and I'll see you guys in the next video on pathology of tuberculosis. Right? Thank you so much for watching.